going to start with host way today. I hope y'all are excited. Thank y'all for being here. I really appreciate that. All right, so host way, if you want to do a quick intro of yourself, I'll let you do that. And the meeting's recording, so you're good to go.
Can I go ahead and share my screen?
Yes, but
Okay.
let me know before you start presenting.
Okay, for sure. So good morning, everyone. My name is Josue Zuniga. I am a senior biology major at Tarleton. Can everyone hear me okay and see my screen? Okay, cool. Okay, I'm gonna go ahead and start right here. So I work with the parasitology lab at Tarleton, and my research is titled The Diversity of Phlogistomum, classified through CO1 and ITS. And so before I get too deep into this, I just want to give a quick overview of what Phlogistomum are. Phlogistomum is a trematode genus, and trematodes are basically parasitic flatworms that are internal parasites of mollusks and vertebrates. And these parasites utilize a three-host life cycle, with herons being their definitive host. However, we're most concerned with the stage of its life cycle where it's found inside of its secondary hosts, which are freshwater fish. And in this stage of its life cycle, they're commonly known as white grub. And these white grub have been reported from several tissues of multiple different species of freshwater fish. And they've actually become a concern for fisheries due to the infections that they cause, which have detrimental effects on the host health. And so this is because the way that the parasites enter their secondary host is through penetration of their tissues, and they migrate through those tissues as well. And this can lead to mechanical damage, things such as hemorrhaging, secondary bacterial infections, and in many occasions they can lead to the death of the fish itself. And a lot of times fish that are also that are infected with these parasites actually become less able to avoid predation by their avian, the herons that feed on these fish. So here's a brief overview of the life cycle of these parasites. So their adult stage is in the herons, and herons actually live in the intestines. And so these adults release eggs, which are then released by the heron through its waste. These eggs enter the water where they're taken up by snails. And in snails, these eggs hatch and become cercari. And these cercari mature and they become free-swimming cercari, which can then leave the host and swim through the water, where they then penetrate the intermediate host, which is freshwater fish. And like I said, they penetrate their tissues, and they can migrate to areas such as the liver all the way up to the eyes. And these can cause detrimental effects on their health, like I mentioned, especially in the liver, where they can form a lot of cysts. And whenever these cercari mature inside of these freshwater fish, they become metacercari, and they encyst. So they form these cysts in the tissues. And once again, these parasites can then be taken up again by their definitive host whenever they prey on these fish, and these parasites leave their cysts. So over 25 species of Phlogistomum have been described in different parts of the world. However, the life cycle and the host species are only known for actually like 4 species. And this is because they've proven to be exceptionally difficult to identify morphologically at their larval stages, due largely in part to a phenomenon known as cryptic diversity, in which species share morphological characteristics, but they differ molecularly. And so scientists are now having to rely on molecular-based studies in order to correctly identify these species. And so without correct species identification, we can't really determine the host specificity of members of this genus. And host specificity is basically a measure of the diversity of hosts that a parasite can infect at a particular stage in its life cycle. And this degree of host specificity has a direct effect on the ability of a parasite to infect other hosts should its primary host become unobtainable. So that's kind of where our research comes into play. So our goal here, given how widespread Phlogistomum infections are within freshwater fish, it's important to understand the infection dynamics of this genus. And like I mentioned, these trematodes can only be accurately identified molecularly. So our research is meant to determine the diversity of Phlogistomum in larvae, in their larval stages in various different fish hosts in Texas. And so we're doing this through the phylogenetic analysis of molecular data. Now, 2 molecular markers have been successfully used in order to identify eukaryotic species at all developmental stages. And these would be the mitochondrial cytochrome oxidase 1 gene or unit, which is CO1, and the internal transcribed spacer gene or ITS. And these 2 markers were chosen specifically for this study because they're the most commonly used in trematode systematics. And we chose to use 2 different markers instead of 1 for a more accurate identification of these species. And then aside from that, CO1 and ITS sequences are also readily available on GenBank for these trematodes, which is where we drew a lot of our species sequences from to compare our isolates to. So how did this research go about? Well, we obtained isolates from various tissues of host species, like I said, ranging from the liver to eyes, even ovaries. And here's a picture of an encysted eye that has various postal-diplostomes inside of it. And so we dissected these, and then we moved on to DNA extraction and then to PCR. And initially, we were having trouble moving forward with this project simply because we weren't able to find the correct primers for the CO1 gene. And so ultimately, we had to sequence the entirety of the mitochondrial genome of one of our isolates. And using that obtained mitogenome, we sequenced primers for the CO1 gene in order to use for PCR. So we sequenced the ITS gene and the CO1 gene, and then we sent those off. And then we got those nucleotide sequences back. And so what do we do with those nucleotide sequences? Well, that's the portion of the study that we're at right now at this point in time. And so we're doing phylogenetic analysis of these specimens, and we're using maximum likelihood and Bayesian analyses in order to study these the diversity of these trematodes. And these 2 types of analysis were chosen based on their usage in other studies performed on trematodes. And in order to compare our sequences, we grabbed a lot of sequences from GenBank. And you're going to see that here in the next slide where I have our tree. So these are preliminary results. This is our tree for the maximum likelihood test of CO1. And I want to start off here at the bottom portion of this tree where you can see all of our outgroups. And these were just isolates that were obtained from GenBank that have no similarity to our gene of interest or to our species. And then up next, I want to draw your attention up here to the top portion of this tree, where you can see the most obvious grouping of isolates. And these represent larvae collected in our study from Gambusia affinis, which are also known as mosquito fish. And you can see that all of these members actually fall within a single lineage, which is distinct from the postdiplostomen and ornithodiplostomen clades, which is pretty significant. Now, below this clade, you can see another grouping of our isolates. These are labeled CVs, as well as some of our P isolates, but these are also from the same species. These come from black-tailed shiners. You can see these paired together with a postdiplostomen species that we grabbed from GenBank. And this one that came from GenBank is actually also from a type of minnow. Now below this, you'll see another grouping of our CVs, which are from black-tailed shiners, except these are grouping in a different clade, which is strange because it might suggest that it actually represents a separate clade, even though these are from the same species. However, we're still in preliminary phases of this research portion, so we're hoping to clear that up through the analysis of our ITS gene, as well as another gene that we want to sequence as well to further identify if this is actually a separate clade. Now below this, you'll see a grouping of 2 of our isolates, P137 and 139, which came from largemouth bass. And these are grouped together with 2 species that we obtained from GenBank that came from smallmouth bass that actually came from all the way up in the Great Lakes region. And so finally, down here at the bottom left, you'll see 2 of our isolates, which came from bluegill, which is a type of sunfish. And these are grouped together with three sequences from GenBank that all came from a Centriarchidae, which is a parasite that's specific to Lepomis, which is it's a family, well, it's a sunfish genus. So you can see that those group together. Now, like I mentioned, we're currently in the process of performing our maximum likelihood analysis using our ITS sequences. The ones that I showed you were all CO1. And we don't really have a good ITS maximum likelihood tree to show you yet. However, preliminary results are showing a lot of similarities to our maximum likelihood for CO1 in regards to the clades that are formed, as well as the host associations. So what conclusions can we draw from our research thus far? Well, we can't say that all our conclusions are solid yet because we don't have enough molecular data to support our claims. However, we are seeing a distinct lineage being formed by our mosquito fish isolates that they're forming a distinct lineage that's different from the Poecilodyplostomum and Ornithodiplostomus clades. And as a matter of fact, we have another student in our research lab who is conducting research on the same species, and she's found an adult stage of one of our isolates that forms part of this distinct lineage. And thus far, we haven't found, or she hasn't found any species descriptions that it matches. So it may represent a new species, but that's entirely her research. But our research is kind of helping her try to identify whether that does represent a new species or not. We also noted that some of our minnow isolates group together apart from the rest of our isolates, like I mentioned in our tree, and that may represent a different clade. And we hope to verify this through the analysis of another gene, as well as through our ITS phylogenetic analysis. Most importantly, however, our current analysis shows a very high degree of host specificity, which supports results reported by other studies that have been conducted in northern North America. So what's next for this project? Well, first and foremost, we want to finish up our Bayesian analysis for the CO1 gene, as well as start both of our phylogenetic analyses for our ITS gene. And results from these analyses they'll solidify our observations, and they'll allow us to make conclusions. And finally, we also wanted to incorporate molecular data from the 28S gene, which is a highly conserved gene. This wasn't part of the original plan, but we're wanting to use this for a couple of our isolates in order to clear up some of those discrepancies that we're seeing in our phylogenetic tree. So lastly, I just want to give a quick thanks to the Southwestern Association of Parasitologists and the College of Science and Technology here at Tarleton for their funding of this project. I want to thank all the students who came before me, because this has been a long project. This has been like a 4-year long project. It's been ongoing. And I want to thank all the students who helped in specimen collection, as well as preliminary genetic lab work. And then a special thanks to Dr. Fow, Carrie Waddle, and Dr. Blasco Costa for all their help in things such as genome skimming, as well as for answering some of the many questions that have come up throughout this research. And finally, I just want to leave some time for any questions that you guys may have.
Does the parasite also harm the other hosts like the snails and birds, or does it just hurt the fish the most? Is that why it's of the most concern?
So yeah, the most concern would be for freshwater fish, simply because that stage of their life cycle, they cause cysts in the tissues, which can be very damaging. But otherwise, in herons and in snails, they pass through their intestinal system, and if anything, they may have an effect on the way that they eat. They may, because they feed off of the same food that they eat. But other than that, they don't have many detrimental effects on the primary and the next host as they do with the intermediate host.
I have a question. Yeah. So I think the flow of your presentation information was pretty clear. Just I would like to can you help me to understand something? So you mentioned like the host specificity, this word several times. So does that mean like this parasite, they preferably choose certain species as their host? Is that what that means?
Yeah, so we're trying to identify the host specificity. So what hosts they, like if a species of parasite only infects a specific host, or if it can branch out and infect other hosts. So for example, can the species that we grabbed from the minnows, can they also infect species that are, or hosts that are from the sunfish species? That's what we're trying to figure out. But so far we're seeing a very high degree of host specificity. So those that are infect minnows only infect minnows, those that infect sunfish only infect sunfish, and same for bass. But we are noticing a kind of like an anomaly there where with our, here I can go back to our, this slide here. These came from black-tailed shiners, and these grouped together with a black-tailed shiner species that came from a minnow species, and these are minnows. However, these are grouping together separately from any of these other isolates that we've grabbed from GenBank. So this may represent a different host, the same species infecting a different host. But that's what we're trying to figure out. But like I said, this is all very preliminary, so we're barely starting this phylogenetic analysis.
Yeah, I think this is pretty cool. Like, if once you have a more completed results, you
right.
Yeah, you could presumably like you have those species identified. You could further maybe study those freshwater fish, like study those habitat. What kind of environment they primarily like live at. So I think you could have a very very deep research. Like you could review
exactly.
information.
Yes, and
yeah.
And this is one of many trees that we have planned out. This is only maximum likelihood for our CO1 gene. So we still have to do Bayesian analysis for our CO1 gene, and then we have another gene, the ITS gene, which we're going to do a maximum likelihood as well as a Bayesian analysis. So we're going to have a lot of data to support our claims, hopefully.
So when you got these samples, you took them directly from minnows? Is that for doing the genetic analysis?
Yes. So I actually didn't. So like I said, this has been an ongoing project, and it was actually on hold until I started this last semester. So all of these specimens were already obtained. I just moved forward with the DNA sequence. I mean, with the extraction and the PCR. And so from what I know, they took these specimens directly out of the tissues of the fish.
Do you know kind of like were they just kind of all across the country? Because I know you said some of them were more Great Lakes focused.
Yeah, well, that was for the specimens that we grabbed from GenBank. These two, they're smallmouth bass, and those came from the Great Lakes region. But all of these fish were collected in Texas. I'm not sure exactly where, but they were all from Texas. Yes, ma'am.
Okay, so the phylogenetic tree is kind of like a mix from ones that were directly collected and the gene bank.
Right, yes.
Awesome.